Microbial Culture Collections for the opportunity to co-partner in this relevant and timely symposium. My congratulations to the organizers for the success of the symposium, and a good day to all. Stay healthy and safe, everyone. Thank you very much.
Thank you very much, Dr. Luna. And okay, so um, thank you. We will now hear the opening remarks from the current president of the Philippine Network of Microbial Culture Collections. She's a graduate of Master of Science in Microbiology and a registered medical technologist. Currently, she is a member of the faculty of the Department of Medical Technology of the College of Allied Medical Professions of Angeles University Foundation. Ladies and gentlemen, PNMCC President Ms. Diane L. Dizon.
Good morning to everyone. On behalf of the Board of Directors of the Philippine Network of Microbial Culture Collections Incorporated, it is my privilege and pleasure to welcome all our distinguished guests to the 20th PNMCC Annual Symposium webinar series. PNMCC is committed to lead in the standardization and upgrading of procedures for the isolation, characterization, distribution of microorganisms, and biosafety to training and seminars. Our task is to provide awareness and give emphasis on the importance of culture collections, especially in this time of pandemic. As the network marked its Platinum Symposium anniversary, PNMCC chose the theme 3Cs, which stands for Culture Collection and COVID-19. This year's annual symposium will be very different from the past symposiums because it will be held virtually, and it will be comprising of webinar series which will be held on the 4 Saturdays of October. The webinar series will address issues and concerns regarding the very controversial COVID-19, giving highlights on topics related to testing of the virus, updates on the development of vaccines, the role and importance of biosafety and biosecurity in the laboratories, and the urgency for the establishment of the Virology Science and Technology Institute of the Philippines. As for the first day of the webinar series, we invited prestigious speakers who are going to share their expertise and knowledge tackling on topics on the relevance of culture collection, on human health under the current situation, the presentation of the different testing platforms in the aid of COVID-19 diagnosis, most specifically on the nucleic acid-based testing, and the science behind rapid testing. Truly, these topics are very significant and timely in this global crisis we are facing too. I sincerely thank our honored speakers for sharing their invaluable time and effort with us. I also want to thank our dear sponsors for their generous contribution and support to the network. To the administration, faculty, and staff of the Trinity University of Asia College of Arts, Science, and Education Biology Department, thank you very much for collaborating with us by granting our requests to use their Zoom platforms and social media accounts in the promotion and presentations in the webinar series. We look forward to more collaborative work in the coming years. And to the undisputable board of directors of the PNMCC, my sincerest gratitude for the dedication and hard work in making this event a success. With that, I welcome again our dear participants and guests. May you gain extensive knowledge with a feeling of gratification for participating in this webinar series. Thank you and a pleasant morning to all.
Thank you very much, PNMCC President Diane L. Dizon. And before we proceed to the presentations of our esteemed speakers for this morning, we would like to remind our dear participants with our webinar guidelines. So the 20th PNMCC Annual Symposium webinar series has the following house rules. First, please carry yourselves in a respectable manner throughout the webinar. Secondly, to be understood by your speakers and other attendees, especially those who are coming from other countries, everyone is encouraged to speak in English. Thirdly, turn off your cameras and your microphones while speakers are presenting. And this will lower the bandwidth and prevent background noise from disrupting the webinar. Fourthly, you are discouraged, or rather you are encouraged to send your questions and concerns through the chat box, which will be screened by our chat facilitators. And lastly, due to time constraints, only a few questions will be entertained. We would also like to acknowledge our hardworking committee who are serving as chat moderators in our Facebook and YouTube simultaneous live broadcasts. Uh, for our Zoom, we have Ms. Daisy Sucaldito and Professor Romel Agbayani from Trinity University of Asia. For the Facebook Trinity Talk, we have Ms. Hazel Yahweh Arce from TUA. And for the PNMCC Facebook, we have Mr. Nick Sean Tabao and Mr. Eldrin Argelies. I will now introduce our webinar moderator. Professor Joel C. Cornista is presently an assistant professor of microbiology at the Biology Department, University of the Philippines, Manila. He was the former chair of the Department of Science, College of Arts and Sciences of Miriam College in Quezon City, and a senior lecturer in biology at the Ateneo de Manila University. He is both a registered and specialist microbiologist accredited by the Philippine Academy for Microbiology. Presently, he is the president of the Philippine Society for Microbiology. He is also a fellow and currently the country ambassador to the Philippines of the American Society for Microbiology. He was the former president and currently a member of the board of the Philippine Network for Microbial Culture Collections. And he is also an active member of several reputable scientific organizations. Mr. Cornista holds a diploma in international postgraduate course in microbiology from the Department of Synthetic Chemistry and Biological Chemistry, Graduate School of Engineering at Kyoto University in Japan under a UNESCO scholarship. He was also a research student at the Material and Life Sciences Graduate School of Engineering at Osaka University, Japan. He finished his Master of Science in Microbiology from the Institute of Biology, College of Science, University of the Philippines, Diliman, and earned his Bachelor of Science in Biology also from the same university. He has published several papers, local and international refereed journals, and has presented his research in various symposia, conferences, both local and abroad. His research interests are in the fields of microbial ecology and genetics, and is presently working on molecular gene cloning and molecular diagnostics. Ladies and gentlemen, Mr. Professor Joel Cornista.
Thank you very much, Assistant Professor Cantal, for that introduction. Again, I would like to welcome everyone to the 20th Annual Symposium webinar series of the Philippine Network of Microbial Culture Collections. Today, we will be starting our day one of this month-long webinar series with the theme COVID-19 testing, nucleic acid-based testing, and rapid testing. We're very lucky today because we'll be joined by speakers who are pillars of this organization. We will be starting the lecture with our first speaker. Our first speaker is a pioneer of PNMCC and was instrumental in its establishment 20 years ago. That's a hint of probably how old she is, but what I'm trying to say is that she was there in the establishment of this organization as we celebrate our 28 years of existence. Dr. Ida Dalmasio is a professor emeritus of the University of the Philippines. She finished her PhD major in food science, minor in microbiology at the Pennsylvania State University, USA. Some of the awards that Dr. Dalmasio receives are the Distinguished Teacher Award in Biological Sciences of UPLB, Outstanding Young Scientist Award in Food Microbiology given by the National Academy of Science and Technology, Outstanding Microbiologist Award, of course, conferred by the Philippine Society for Microbiology, UP Diamond Jubilee Professorial Chair, UP Foundation Incorporated Professorial Chair, J.R. Velasco Professorial Chair, among others. There are so many to mention. She also held several administrative positions, such as the College Secretary of UPLB, College of Arts and Sciences, Program Leader, Food Fermentation and Feed Production Program of Biotech, also in UPLB, Deputy Director of IBS, Director of Biotech for 2 consecutive terms, Project Director, Technology Transfer and Training Projects on Applications of Biotechnology for Small Industrial Development, a project in cooperation with Mahidol University of Thailand. She was also the Executive Director of the Philippine Council for Advanced Science and Technology Research and Development, or CASTARD, under the Department of Science and Technology and the University of the Philippines Faculty Regent. She also is a member to different national and international committees, being the national focal person for the Philippines, APEC Industrial Science and Technology Working Group, Philippine representative to the ASEAN Committee on S&T, evaluator of basic researches of UPLB, member of the Technical Committee on Biotechnology Research and Development, DE Biotech Program, lead transfer for food microbiology under the capacity building of educators in training and teaching of selected microbiology courses, and a consultant to the OST Cradle Project. She has established several collaborations for capacity building with different universities abroad, such as Texas A&M University, the Purdue University in the USA, University of Cardiff in Wales, also in Zurich, Switzerland, Quebec, Canada, Baltimore and Honolulu, USA, in Australia, even in Jakarta, Indonesia. Our speaker has published more than 150 scientific papers in refereed journals and has presented these papers in several conferences, both local and abroad. She has also published 3 laboratory manuals, namely General Microbiology, Food Microbiology, and Dairy Microbiology. Ladies and gentlemen, it is my honor to present to you our speaker for today who will talk on culture collections and their relevance through time. Allow me to welcome you all to our first speaker, Dr. Ida F. Dalmasio. Dr. Dalmasio.
Okay. Thank you very much for that introduction, and good morning to all participants, to all panelists who are now present here. I'm glad that I'm here to talk to you about the relevance of culture collections through time, and not only for 1 decade, but it will span all generations. Before I accepted this, I was trying to see what topic I can relate to you or to discuss with you, because it is rather a very difficult task for me. But since it reminded me that some people who are involved with culture collection will just say that they cannot compete with others in terms of publication and the results. So I thought of how culture collections and the people involved here will be able also to do some research, do some collaboration, and you will not be left out in the scientific community. So with that, I would like to tell you that throughout the presentation, put yourself as a culture collection specialist and what you can contribute, what your center can contribute for the advancement of science and for humanity and for society as well. Okay, let me now start with the presentation. Just wait. And I'm going to share. Okay. I hope this works now. Okay. Of course, that is the title, Culture Collections and Their Relevance Through Time. And that is my name. That's not very important. So here's the outline of the presentation. I will make a brief introduction about culture collections, including their functions, which you probably already know, and the general guidelines followed in their operation, and the Millennium Development Goals and the possible role of culture collections for this. Some areas of application or use of microorganisms in different areas of life, then the culture collection interventions for a healthy life, which is boosting the immune system, development of diagnostic kits, production of drugs and vaccines, and of course, the suggestions for a more responsive and relevant culture collection center, which I want you to remember all the time. Okay, just for the introduction, culture collections are biological resource centers. Traditionally, they were composed of microbial accessions only. You have yeast, fungi, and then bacteria, and all sorts of those organisms. But more recently, cell cultures have been added to this biological resource center. So you can have also animal cells, you can have plant cells, etc. for this one. The functions of culture collections, which I have heard already, uh, is to gather, maintain, are to gather, maintain, preserve, authenticate, and catalog the holdings that you have. You distribute genetically stable cultures, and emphasizing that, and cell lines. They are used for research, instruction, quality control assays in the industry, and for various biotechnological activities. And of course, you provide public service, for example, like in patent filing. There are important guidelines that are followed by microbial resource centers. You just don't establish and just don't get the organisms or whatever and then have that as a microbial resource centers, because you work within the Convention on Biological Diversity, which was implemented to support the conservation of genetic resources and utilization of biodiversity. You also have to recognize the principles of fair and equitable benefit sharing. You have to comply with certain legal framework and contribute to achieving the Millennium Development Goals. What are these Millennium Development Goals? There are a lot of them, but I have placed here under bold letters those where culture collections will be very relevant. Let me just go to them. First is to eradicate extreme poverty and hunger, achieve universal primary education, promote gender equality and empower women, reduce child mortality, improve maternal health, combat HIV, AIDS, malaria, and other diseases, ensure environmental sustainability, global partnership for development. Probably you're wondering how microbial culture collections can contribute to this. So let me go to this very fast. To eradicate extreme poverty and hunger means you have to double agricultural productivity and incomes of small-scale food producers, ensuring sustainable food production systems. And then we're also going to go to the others. Reduce child mortality. How? You can use probiotics, combat HIV, AIDS, malaria, and other diseases. And the intervention of microbiology is to reduce common illnesses through research and development of vaccines and medicines for the communicable and non-communicable diseases as well that primarily affect developing countries. Another one is to ensure environmental sustainability by progressively improving land and soil quality, microbial pesticides, compost, which are composed of microorganisms, and biofertilizers. And then global partnership for development, which is to network and coordinate activities to give improved access to high quality biological resources. High quality. Okay. Now, where are microorganisms used? So, to support the Millennium Development Goals in agriculture, I've mentioned biofertilizer earlier because biofertilizers increase productivity and income of farmers. And so we have already some commercial biofertilizers that are in existence. And I also mentioned compost enhancer. This improves the land and soil quality, and we also have some commercially available compost enhancers, and these are also composed of different microorganisms that have been selected. Now, I've mentioned microbial insecticides, These are environment friendly because they are not harming the other organisms in the environment, but they are targeted. Perhaps you know already Bacillus thuringiensis, which is a very important bacterium where we get the different microbial insecticides. And of course, I'm just going to point here the talong that we have, which is really very difficult to grow. And there's now an intervention of Bacillus thuringiensis so that we can get talong, which will grow to ripening without pests and disease. Okay, another one is silage making. This might not be very important in our country as we are having temperature of having almost the same temperature year round, but in other countries, temperate countries, they would ensure availability of feed supply, especially during the winter months. And so this is a method of preserving feeds using microorganisms. So you'll have selections of lactic acid microorganisms for this one. Now, where else? In food and dairy processing, there's a very big contribution of culture collections because we have, for example, the different dairy drinks that are composed of microorganisms to improve the wellness of those who are going to drink that. And then in pickling, and then this is just an example how to make tempeh. I'm sure for those of you who took microbiology, one especially in UPLB, this is part of the exercise that we have that is being made using rhizopus. And then the common fermented foods that we have, you have pickles and other ones which utilize lactic acid organisms. These are the lactic acid organisms. They would have to be selected and that will be the ones that will be applied to these different processing methods. Now, in the industry, one example is ethanol production. You know that you have a selection of different yeasts, which will give you a lot or high volume of ethanol. And aside from ethanol, you have different solvents, acetone, butanol, etc. So you will also have microorganisms in these selected microorganisms. Now, this one in geomicrobiology, the microorganism used is Thiobacillus ferrooxidans, and they are used there to have this uh, bioleach the copper. So this is important also in mining and similar industries. Now, let us have a glimpse of the past and current worldwide health crisis, which are now we are being confronted with. Now, as early as 1200 BC, diseases which created havoc have been reported, though some of them did not cause numerous deaths. But I will just go through some of them. The plague. This is, I think, in Tagalog term is peste. You have bubonic plague, typhus plague, or plague caused by typhoid bacilli. The influenza, who can forget the 1918 influenza pandemic, which killed a lot of people. And then there is a series of cholera pandemics. Then, more recently, which comes into memory, are the HIV/AIDS, the MERS-CoV, and Ebola. And the latest threat to global health is the COVID-19 pandemic. Now, microbiology intervention to ensure healthy lives, just to go over, is we reduce or prevent illnesses, or microbiology can prevent or reduce illnesses through strengthening of the immune system. So there are microorganisms for that. Research and development, the microbial culture centers can do that for the development of diagnostic kits, vaccines, and medicines or drugs for the communicable and non-communicable diseases that primarily affect developing countries. These are the current and time-tested approaches to fight diseases. First, you boost the immune system. We have been hearing of that. You have to be healthy. Now, The contribution that I can think of microorganisms are the probiotics, which I have mentioned before. Just to go over what probiotic is, it's a consumable product that contains live organisms that are or believed to benefit the consumer. These live microbial cells, when ingested in certain numbers, exert a valuable effect on health and wellness by improving the balance of good and bad organisms in the intestine, and the foodborne pathogens are also inhibited. Now, the probiotic cultures that are added to products may be only one kind or different kinds of microorganisms. Usually, they are lactobacillus, such as this one, or bifidobacterium, such as this one. As how they look under the microscope. Now, what are the beneficial roles of probiotic microorganisms that you can supply? They will exclude intestinal pathogens. They stimulate immunity so that the people will be able to better resist infection. Other effects: lower swelling due to gas production. They aid in food absorption and digestion, and also they help in the synthesis of vitamins. They promote lactose digestion in lactose intolerant individuals. This is very prevalent. They prevent establishment of pathogens. They shorten the duration of and enhance recovery from diarrhea, make the conditions in the intestine unfavorable for pathogen, and it has been reported that it might be anti-carcinogenic. Reduced activity of ulcer-causing Helicobacter pylori, which is production through the production of inhibitors by the bacteria in fermented milk, and through immune system modulation, which will strengthen the defenses against infections and tumors and lowers the inflammation. The second intervention is to develop fast and reliable diagnostic systems. Diagnostic microbiology deals with the confirmation of the suspicion of infectious disease and the identification of the etiologic agent. With molecular diagnostics, antibody and antigen tests, microbial cultures are used as research tools, so we're still needed for this one. Microorganisms are needed for strain diagnosis of pathogens, and thus it will depend upon a supply of authentic and viable cultures of these microorganisms which you can supply. Now, the use of biosensors, which makes use of enzymes derived from microorganisms, have also been used in medicine. Therefore, culture collections serve as data source for the development of new diagnostics, and you have a very good role to play in this. Now, the third intervention, drug development and testing of antimicrobials, are compounds which can stop or deter the infectious agent or to reduce their load. Now, collections often maintain novel microorganisms which await future exploitation for drug development. So you have a bank of these novel microorganisms. As in the design of new diagnostics, culture collections serve as data source for the emergence of new treatment. Herbal medicines, as you have probably have heard, against coronavirus has been tried in cell culture model. In vaccine production, viruses are grown in cells or continuous cell lines, and recombinant proteins are manufactured in this bacteria, yeast, or cell culture. And as resource centers, while providing research support during an epidemic, culture collections also provide information as to the toxin production potential of the organisms being used and certain biosecurity risks if there are any. Now let's take a brief look at the current scenario. Okay. The field of microbiology is assuming an increasingly molecular emphasis. We cannot deny that. But the need for culture collections has not diminished since microbes provide tools for use in molecular biology. Culture collections still provide a significant degree of continuity with the past through the preservation and distribution of microbial strains described or cited in publications. Applications in microbiology and biotechnology depend upon a supply of authentic and viable cultures of microorganisms. Okay, molecular, genetic, and whole genome sequence analysis of contents of microbes and other living resource collections bring additional value to living collections. Hence, we have re-emphasized the value of collection holdings. Microorganisms have already proven that they can function as powerful medicine factories, such as insulin for diabetes. Now, these are my suggestions for a more responsive culture collection center. First, you have to invest on capacity building. We do not mean only equipment, the buildings, etc., but more importantly, on the capacity of the people working in the culture collection center, so that we will be able to cater to more diversified roles and be able to use the state-of-the-art techniques. So it's not just collecting, identifying, maintaining, and giving away. So there will be more diversified roles that you can have as culture collection staff. Second, you have to expand the size and interest of the collection, since the detailed services that can be offered will depend upon the collections that you have. If you expand the size and interest of the collection, you can do very many special testing that will be required by clients and other stakeholders. So you just not say that it is enough, it's very difficult to maintain. Well, I know it's difficult to maintain, but you have to expand the size so you will be more relevant. Now you provide consultancy, contract research, and information services. Perhaps here you will be able to generate additional income. You have to work in collaboration, not in isolation. You have to communicate with other stakeholders. Another suggestion, be proactive, not only during microbial outbreaks, but in all public health measures. Now, let us look forward. Based on the records, it is prudent to say that there will be another outbreak. So let us be ready. We're not hoping for that. We do not really like that. But based on the scenario that I showed you before, there have been other pandemics. So let us be ready. Therefore, let us continue research so as to sustain the efforts and contribute to preparedness when their time comes. We have to gather as much information regarding the characteristics of the accession. So if you have a particular microbe or the cell that you have to prepare a detailed characteristics of this, where they can be used, what can be made of them. That can be used for developing strategies to combat future outbreaks. This is my caveat. Microbes don't care about your economic status, your culture, or your politics. Microbes only care about reproduction, spreading, and finding the next host. If the quarantine measures are relaxed, the virus does not relax. Microbes don't need our textbooks, our public health plans, nor do they listen to experts. So now that we are facing a very grave pandemic, all of us are susceptible to this, but we have to be careful. But aside from just being careful, we have to spread out our intelligence, our knowledge, or our capacity on how we can contribute to this. Mind you, you can contribute a lot, not just by being careful, but by practicing the health suggestions like washing your hands often, wearing your face mask, wearing your face shields, avoiding crowded rooms, avoiding public places where there are a lot of people. But here you have to be more relevant. The global community must work together to reduce and defeat these outbreaks. This has been done before and we can do it again. We who are involved in culture collections must do our part. Before I end, let me just tell you again that you play a very important role in the global community, not only by giving the microorganisms or the cell lines or characterizing them, maintaining them, but we have to work together in order to find out what we can do, what we can research on, what will be needed by the other stakeholders. So you are a very important part in the scientific community. And so please maintain that you are being a very good science worker, a researcher, and also an extension specialist. With that, I end my sharing, and I hope you will be more enlightened on how you can contribute to the pandemic solution, to the solution for this and for all other health emergencies that may come our way. With that, I thank you and I hope that you will still be very, very active in cultural collections and in participating in all health problems, and not only in health, but other sectors of society as well: agriculture, industry, food processing, and other related endeavors that will require your expertise. Thank you very much and good morning. Mabuhay tayong lahat. Mabuhay ang culture collections. Okay. Thank
Thank you
you.
very, thank you very much, Dr. Dalmasio, for that very informative lecture. Okay, the floor is now open for questions. For participants, you can write your question in our Q&A box located on the lower part of your screen. And for those who are watching us in YouTube live and in our FB page, you can forward your questions to our moderators. Okay? So, may I start with the question to Dr. Dalmasio? What is the significance of a culture collection to a country like ours, the Philippines? And where do you think we are in terms of collections compared to our neighbors? And in the last 20 years, what do you think has been the improvement in our culture collection facilities in the Philippines?
Okay, for the first question, that the relevance of culture collection in the Philippines, it is very relevant because you have microorganisms in our accessions that can be used especially in agriculture. You know that we are an agriculture dependent country at the moment, and so we need microorganisms in order to help us in growing the different crops that we have. But the trouble is sometimes there are people who do not realize that. But we have to be more proactive and we have to tell them the importance of microorganisms. And then the other question which I can remember is how we fare compared to our neighboring countries, right? Okay, by neighboring countries, we can probably compare ourselves to other Southeast Asian countries, but you know that there are other Southeast Asian countries which do not have, which I would say would not have as many collections as we have, and probably you know already the countries. However, for certain countries, our neighbors like Thailand, Indonesia, well, Singapore, I would say that, and Malaysia, we can compete with them. We can compete with them because in terms of our capability, we will not let ourselves be cut short by their capability. We have the capability. Only let us be proactive. Let us ask the government to give us more support, and we will be rising to the challenge. I think those are the questions. Okay.
There's another question from our Zoom from Maria Filicana Benita Iloreta. Is there a limit to take in probiotics or can it be a maintenance supplement already?
Okay. Very good question. There is actually a certain level of probiotics that should be circulating in your body. It should not be lower than 10 to the 8th or 10 to the ninth circulating. And you have to maintain that. That's why your commercial establishments or the factories would say that you have to take this regularly because you know that microorganisms have certain span of life. So if you do not maintain them, they will tend to die down with time. So you have to be very religious in taking them. So that is my advice for that. But there is a certain number, 10 to the 9 per ml at least. But the commercial ones, they contain a lot of them. So when you take them in, you will have that, but you have to continue taking them in so that microorganisms will be replenished. Okay.
Okay, thank you very much, ma'am. We have few more questions here. Another one is, what is the role of culture collections in publishing new species of microbes and patent application of biotechnologically important microorganisms?
Okay. For um, the staff or the researchers that are involved in culture collection, you can do your own research for the organisms that you have because you cannot just depend on the depositor for the characteristics of the organisms that they have. So you have to verify them. And in so doing, you might be able to stumble on some characteristics of the organisms and you can work on that. You can continue that. Initially, you're trying just to determine the characteristics, but so doing, you might be able to continue with the research on the particular organisms that you're interested in. And now, with regard to other publications, of course, you cannot do that by yourselves because there are many researchers who are also using that. So you have to collaborate, you have to seek permission, or you have to make sure that what you are doing is also relevant to the research. And when you come up with certain results that are worth publishing, you have to collaborate with the researchers that initially worked on this. So that's what I meant by collaboration, not in isolation. For patent filing, of course, you cannot just file a patent for certain microorganisms or the processes. We have to have the culture collections must maintain the microorganisms stable cultures because in patent filing they will be coming back to you because there will be some questions that will be asked. So the culture collections should be able to provide the organisms that are being used for patent. That's it.
We still have a lot of questions in line here, but we could probably can answer one more. And the other questions, we will be forwarding it to you, ma'am, and then we will probably informing those who ask the questions for their specific answers. So the last question
Okay.
that we have for today is, what specific training, consultancy, and contract research and information service can we avail from culture collections in the Philippines?
What I am thinking here is that I have been when I talk to different science organizations or to different professional organizations, they're interested in consulting with what can I use for this process that I am intending to do. This is especially true for the students who are doing their thesis. So I advise them to seek the culture collection and ask them what organisms they can use for that. So that is one way of consultancy. Don't think initially of very big consultancies. As government workers, we have to be of service to the students, especially who are going to follow our footsteps. And then for aside from consultancy, what was the other one, Joel?
Uh,
Consultancy.
uh, Consultancy, contract research, and information service.
Oh, okay. The research and information service, well, the research, probably if you can only have a list of microorganisms that you want to have, you already have that, then you can add the culture collection additional information that they have on this particular organism. But please bear in mind that different organisms will have different strains and their capacity for microbial whatever metabolism or whatever biochemical processes might differ in different microorganisms depending on strains. So those are just what I can think of at the moment.
Last na lang, ma'am. There is a very important questions and very interesting question from Dr. Wilfred Chu. Do you think the bioprospecting regulations limit our effort to increase collections? Regulations, bioprospecting regulations.
Okay. With regard to that, we know that there is an EO regarding that, the bioprospecting, right?
Yes,
That you before you are able to get your samples, you have to get the permission of the mayor of
permission from
whoever.
different departments, yes.
Yes. Somehow, when I was still working, somehow it became limiting for me when I was doing that. However, what I do now, what you can do is before you do that, you just have to write, although this is another layer, you have to write and you have to seek permission. Somehow, instead of us just gathering, is really it deters us somehow, but this should not really be a wet blanket for you. But we have to find out ways by which we can expedite. But this bioprospecting law was enacted so that the community where you got your organisms will also benefit. So somehow, but let's try to do something about it. You can do something about it anyway.
Yeah. I'm very sorry, due to the lack of time, you would need to cut the question and answer, but there are a lot of questions in our Q&A box. Maybe Dr. Dalmasio can look into that later. And we will just try to forward to you some of the other questions coming from YouTube, because we also have
Yes,
participants watching us from
yes,
YouTube. So
yes. I
at this
yes.
point, I would
Let us
like,
just for the questions, please send them to me through email. And so
mhm.
for those who send questions, please also give me your email address so that I can respond to your question. Thank you very much. Thank
Okay,
you,
thank
Joe.
you very much, Dr. Dalmasio. At this point, I would like to give our certificate of appreciation to Dr. Dalmasio for sharing, okay, uh, 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 all of her thoughts regarding culture collection. Okay, so on behalf of PNMCC, thank you very much, Dr. Dalmasio, for gracing us as a speaker in this 20th annual symposium event webinar series of PNMCC.
Thank you. Thank you. Salamat. And be proactive. Thank you.
Okay. Thank you very much. At this
Welcome.
point, we will be moving on to our second speaker for today. Okay. Our second speaker for today is Dr. Lourdes M. Nadala. She is also another pioneer of PNMCC, instrumental in the establishment of this organization way back 20 years ago. Dr. Nadala obtained her BS Biology degree with a major of microbiology from the University of the Philippines, Los Baños. She worked as a research assistant at the culture collections of the Museum of Natural History in UPLB under the tutelage of the late Professor William L. Fernandez. She pursued her MS in microbiology degree also at UPLB and then became the head of the Philippine National Collection of Microorganisms, PNCM, at Biotech, and along with our first speaker, Dr. Dalmasio, they started networking with other culture collections such as that of NSRI, ERI, ITDI, and MNH. Dr. Nadala obtained her PhD in microbiology in 1996, and upon her return to UPLB, she collaborated with the Bureau of Fisheries and Aquatic Resources, or BIFAR, to help alleviate infectious diseases problems in aquaculture. In the year 2000, Dr. Nadala pursued his postdoc research at the University of Cambridge in UK on a viral discovery project on chlamydial diagnostics. In 2002, their laboratory at the University of Cambridge established a spin-out company in the Bay Area in San Francisco, California, called Diagnostics for the Real World Ltd, or DRW, which she is connected also up to this time as vice president. Dr. Nadala initially worked as a scientist assigned to develop an immuno-based assay for HIV for the use of resource-limited setting. She took a break from human diagnostics and worked at the Molecular Epidemiology Incorporated Institute of the Environmental Health Incorporated in Seattle, USA for 3 years. After 3 years of hiatus in the human diagnostics, Dr. Nadala is now back in DRW as Vice President for Regulatory Affairs and Quality Assurance. Dr. Nadala has co-authored numerous articles published in refereed journals or presented these papers in scientific meetings and chapters in a book. She is a life member of the Philippine Society for Microbiology, Sigma C Research Honor Society, and is a diplomate of the Philippine Academy for Microbiology. Ladies and gentlemen, to talk on SARS-CoV-2 testing platforms in aid of COVID-19 diagnosis, let us all welcome Dr. Lourdes M. Nadala, the Vice President for Regulatory Affairs and Quality Assurance, Diagnostics for the Real World Ltd. Dr. Nadala.
Hi, thank you so much for that introduction, Professor Cornista. And I would like to thank the organizers of this symposium. And it started on a Facebook post. And I was very happy to hear that the PNMCC, or the Philippine Network of Microbial Culture Collections, has sustained from 20 years ago. It started as an informal group of 4 culture collections, which I heard now has grown into a bigger and more wider, with wider membership throughout the country. So for today's talk, I was asked to talk about the testing platforms on SARS-CoV, having been in the diagnostic, working in a diagnostic company for the last 20 years. And I'm sorry, but there's been a glitch. So I have asked Carlo to show my slides. So this morning, I will be talking about the different testing platforms for SARS-CoV-2 in aid of COVID-19 diagnosis. So just to limit my talk, because I'm given only 30 minutes, I have prepared this outline. I'll try to give a brief introduction about the target, which is the SARS-CoV-2 virus. And then what is the intended use of diagnostics? Is this for active infection, or do we want to know prior exposure in asymptomatic, pre-symptomatic, and symptomatic individuals? And then what are the base technologies for the current diagnostic platforms? Most of them are nucleic acid amplification based, and they are called the molecular tests. And there are some also which are immunobased, which is looking for antibodies against the virus or antigens. And I would talk more about the applications of these two nucleic acid amplification based tests and the immunobased rapid tests. And I would like to talk a little bit about product approval and authorization, because it's very important that diagnostics are regulated to make sure that we have good quality tests to avoid false positives or false negatives in high frequencies, because there's no 100% sensitive or specific test. But at least we should have some way of making sure that the tests are sensitive enough to detect the target pathogen. And then lastly, which is the most important part of my talk, is the role of culture collections. And I'm very glad that Dr. Dalmasio, Ma'am Ida, has already talked a lot about the role of culture collections in this pandemic. And so I will be talking more about role of culture collections in providing reference materials, which is very important when you are developing a diagnostic test. You have to have a very well characterized and very well quantified reference material, so you're able to know your limit of detection, for example, your specificity or your sensitivity, and then panels for proficiency testing. Dr. Dalmasio has talked about earning some income for the culture collection, and this is important for sustainability, because I know when I was working there, we could not survive on the budget alone from the university. So we had to diversify by providing services to the university and other academic institutions, to industry. And we were also providing consultancy, as Dr. Dalmasio has mentioned. But I think now there's more regulations or in the university. So that's another topic for discussion. And of course, the major role of culture collections as repository for production strains. So most of the molecular tests are using enzymes which are either produced as a recombinant protein, for example, or a recombinant enzyme by bacteria. So it is important to keep them stable, and so that you don't have a drift or a shift in your activity of enzymes. Next slide, please. So the SARS-CoV-2 virus belongs to the family Coronaviridae. And I have a picture here, which is from the National Institutes of Allergy and Infectious Diseases. It's a very stunning picture of an electron scanning micrograph. You can see the spikes, which look like a crown. So that's why they are called coronaviruses, because they have this very distinct spikes on their envelope. It's an enveloped virus which has plus strand RNA, meaning to say that the RNA can be used as a template right away for replication or for amplification. So they have one of the largest, if not the largest, genome. And I would like to emphasize the envelope viruses are easily uh, inactivated by detergents. That's why the WHO has been uh, uh, asking everybody to wash your hands as frequently as possible with soap and water. And they replicate by a unique mechanism, which is they call it as discontinuous transcription and polymerase jumping. So because of this mechanism, it results in high frequency of recombination. So we are in an early stage of the pandemic, so there's still a lot of unknowns about the virus, but in general, the family or members of this family are known to have high frequency of recombination, and we have to keep this in mind when we are developing vaccines. So there are 5 other human coronaviruses, and the last two, SARS coronavirus and MERS, were the ones that we heard of in the last few years. The others did not cause that much impact as far as public health is concerned. But SARS was sort of localized in Hong Kong and some parts of China and Asia. And MERS was it was also localized mostly in Middle Eastern countries. And of course, now we have the SARS-CoV-2, which has created a pandemic. And it's highly contagious compared to the other two relatives. And also because of migration and traveling during the Chinese New Year, that sort of really triggered the full blown epidemic here, especially here in the US, because in the Bay Area there's a lot uh, of Chinese or a Chinese ethnicity, so they would always go back to China or Hong Kong during the Chinese New Year around February, and so that's how it got really widespread and transmitted very fast here in California. Next slide, please. So just to give you the most recent, as of September 30, the global number of cases have already reached over 33 million. And number of deaths have reached over 1 million deaths. So in the US, we have over 7,000 cases, and deaths, which were mostly in New York, already surpassed 200,000. And the good news is we had also a good number of recoveries. In the Philippines, we have been also watching the pandemic, and the cases have already breached the 300,000 mark, and deaths over 5,000. And good news also is we have recorded 253,000 recoveries as of September 30. So what are the current diagnostic tools for SARS-CoV? I still remember we were supposed to go home to the Philippines in February, and then this news broke out, and our CEO said, Lou, can you please postpone your trip because we have to develop a test in as soon as possible, because there was no diagnostic test available at that time. So everybody in our group, Cesar, and our team in Cambridge had to work like round the clock to be able to develop a test in like 6 weeks. So we needed something that is extremely sensitive, but at the same time, it should not be giving us a lot of false positive results. And secondly, it should be able to detect active infection. So uh, since it's a very highly contagious pathogen and also very fatal, you want to detect it as soon as possible. So you want to detect for the virus itself, not antibodies, because antibodies are formed much, much later during the phase of the infection. So, and then when you're developing a diagnostic test, you have to think of the specimen types, or what are the most targeted organs of by the virus or by the pathogen. So, of course, SARS-CoV was known to cause uh, respiratory diseases. So we started looking at nasopharyngeal swabs or specimens, or oropharyngeal, which are very uncomfortable to collect to the patient. And then England use a combination of nasal and throat swab as specimens. So what they do is they're given a swab, and then use it to collect first from the throat, and then collect from the nose. So it was a little bit uncomfortable for us when we had to donate our samples during the assay development. And recently, there have been papers about saliva, which is less invasive, and so they have shown that it has equivalent sensitivity to a nasopharyngeal or oropharyngeal swab specimen. So it's more patient friendly, I would say. So I think now most manufacturers are trying to validate their assays using saliva. And so the most common platform in the field of SARS-CoV diagnostics is, of course, the reverse transcription PCR, or transcriptase polymerase chain reaction. Uh, I think it constitutes about 95% of all the molecular tests on the market. And then there are a few tests like our test, which is based on isothermal amplification system. So as you all probably know, polymerase chain reaction, or PCR, is a series of cyclical amplification in different temperatures. So you have to have a high temperature to separate your DNA, for example, and then allow the primers to bind and then anneal and extend. So that's another temperature. So most of these assays take like 35 to 40 cycles of alternating high-low temperatures. In contrast, NASBA is an isothermal amplification system. So it amplifies only at one temperature. So it's simpler in terms of equipment. So you don't have to have a machine that would heat up to 95 degrees, for example. Then there's the LAMP, or the loop-mediated isothermal amplification. So it's similar to NASBA, but they use different enzyme systems. And then the most recent technology is called CRISPR. Again, this technology is based on either isothermal amplification or reverse transcriptase PCR, but it has a different detection method. So I was trying to look up the Philippine FDA of what are the available tests in the Philippines, and I couldn't find information, so I'm sorry, but most of the presentation or the data I'd be presenting would be from the US and from the UK. And then the other platform that's being used is immunobased, which is what we are, it's commonly called as the rapid diagnostic tests or the RDTs. And for these methods, we are looking for antibodies to SARS-CoV or antigens. As I have mentioned, antibodies are produced by your body in reaction to the virus, and usually this happens later at the later stage of the infection. If you have antibody or antigen-based rapid test, which is also classified under immuno-based methods, that would give you an indication of active infection. But antigen-based tests are very difficult to develop because antigens are not, you cannot really amplify them like, unlike the nucleic acid. So your technology should be very sensitive to be able to pick up small amounts of antigens or viral parts in the body. So the antibody-based tests, although they give rapid results, are not ideal for diagnosis. And I have read a paper from the UPPGH where they compared where they did a performance evaluation on rapid tests being used in the Philippines, and some of them had very, very low sensitivity. So I think there was one which was showing like 20 to 30% sensitivity. So it's worse than flipping a coin, which means that you will be getting a lot of false negatives because the sensitivity is very low. However, having said that, it is also important to know if somebody has already developed antibodies, because you can use the convalescent plasma from that individual who has antibodies for passive immunization of uh, patients who are seriously ill from COVID-19. So this is the basis for using plasma from recovering individuals, because they would have the antibodies that can be used by patients who are extremely ill and who cannot, it's almost like there's no other cure. So uh, the specimen type for this, for the rapid tests, is of course whole blood. So it is a very simple test because you can give your finger prick, a blood from your finger prick, but it is not sensitive enough to detect active infection. So in the US, for example, they are not used to for diagnosis. So they are only used for epidemiological studies or for detecting antibodies in in recovering patients. The antigen-based tests, however, have been authorized by the FDA, and there are quite a few which have shown very good sensitivity and specificity. And I have listed here a link to a website where they have listed the antigen-based tests which have, and also antibody-based tests which have received authorizations from FDA. Take note of the word authorization because of the emergency nature of trying to come up with diagnostics for SARS-CoV. in as early as March, the US FDA relaxed its regulations in order to allow tests to be used for diagnosis. So this was how desperate the government was. But having said that, they still had some criteria that the manufacturers have to meet in order to be able to get the authorization of the US FDA. For example, during that time, there were no cultures or there were no isolates of SARS-CoV. So FDA allowed sensitivity, specificity testing by blast analysis, which is just comparing the sequence against a database of sequences of different microorganisms, different viruses, just to make sure that it does not pick up non-specific targets. Next slide, please. So from here on, I'll talk more about the molecular methods. So this is just a simplified drawing of uh, molecular methods used for the detection of coronavirus, specifically SARS-CoV-2, is, as I have mentioned, RT-PCR and then the isothermal nucleic acid amplification. And there's a whole lot of other technologies, but all of them are based on amplifying the nucleic acid targets by cyclical nature using different sets of enzymes for each type of method. So next slide, please. This is just a simplified drawing of how the RT-PCR works. First, you have your RNA, which for SARS-CoV-2 is already a positive strand. So it can easily be converted into a complementary DNA or cDNA using the AMV reverse transcriptase, or there's other manufacturers use MMLV, which are viruses uh, producing this reverse transcriptase. Next slide, please. Then this is just a simplified drawing of the PCR, where you have, once you have your cDNA, then you have you expose this to a high temperature, so your double-stranded DNA will separate, and then each of these will become a template for more DNA. So at the end of 40 cycles, 35 to 40 cycles, you would have millions of copies of your target. So at that point, you have increased the sensitivity of your assay, and then you do the detection using either using dyes or using the nanogold. For example, you can visualize the presence of your amplicons on a dipstick, which is now being used in some of the assays like Samba, which is what we have, and also the CRISPR-based technology. Examples of RT-PCR-based SARS-CoV-2 assays are, I've heard about the UP RT-PCR, and of course, I think the Gene Expert by Cepheid is being used in the Philippines. And Hologic, CDC was the first one to be authorized. They came out with the first molecular test here in the US, which they have improved and then came out with a version 2. And then, of course, you have the other players like Abbott, Roche, Illumina. So there's about 170 authorized tests, molecular tests in the US, right now, and about 95% of them are RT-PCR based. The next technology is the NASBA, which is what we use for our assay, which is called the Samba2 SARS-CoV-2 test. Some of you may have heard about Samba2 when I presented this at the PSM. Samba2 is a machine which is like a coffee maker in size, but it can be used to detect different targets. So you just replace your reagents with different set of cartridges for HIV. Now we are using it for SARS-CoV, and then we are coming out with a quadruplex, which would be targeting flu A and B, and then RSV, and of course the SARS-CoV. So for NASBA, there are three enzymes that are being used. So we have the single-stranded RNA, and with the AMVRT, the template is being used to synthesize a DNA RNA hybrid, and then the RNase H will cleave this hybrid, and then you have two strands which will become again a template for more amplicons, and this is repeated several times. And so at the end of the cycle, if your target is present, then you will be able to have you will be able to detect the presence of your virus by either by fluorescence or by visualization on a dipstick, or by using other detector probes. Another one I also would like to mention about Abbott. They have also come out with a very simple test, but I'm not saying this against Abbott, but FDA came out with the LOD, or the limit of detection, for a lot of assays here in the US, and Abbott ID now has, based on the FDA evaluation, has like an LOD of 300,000 copies per mil. So maybe they have improved it already, but that's where they are when it was reported last month. Next slide, please. Then you have the lamp, and this is similar to NASBA in the sense that you also have isothermal amplification, so it's only one temperature. But this technology has 4 unique set of primers, which allow the target to, if it's present, would allow to form these dumbbell shaped structures because of palindromic sequences. And these amplicons will form this high molecular weight material, which at the end of the cycle would be visible to the eye. Next slide, please. So you can actually visualize it. So for example, if you have a phenol red indicator, it turns into yellow. So that means it's a positive for your target. Or you can use another, a different kind of dye. So it turns from purple to blue. So LAMP technology has also been used for other pathogens, for detecting other pathogens. And there's a few tests in the US that are based on LAMP. And then the last technology I would like to talk about is CRISPR. So this is just a cartoon of the LAMP technology. Next slide, please. The last one is called CRISPR, and there's one test in the US which is called Sherlock. they're very clever in naming their tests. So it is also based on LAMP or RPA, where there is amplification that is involved, but it uses a certain enzyme where when uh, uh, it detects uh, the target, the uh, enzyme becomes goes crazy and then cleaves the target with the detector, and then this detector emits light which can be uh, read or detected by a fluorescence based detection, or it can be just a chromophore which can be detected visually on a dipstick. Next slide, please. So in essence, all these molecular-based technologies have uh, the common feature of amplification of the target nucleic acid, but the difference lies in the temperature requirements, and then the enzyme requirements, and of course in the detection methods downstream. So this is also based on CRISPR, which is the version 2 of Sherlock. And so, as I have mentioned earlier, there are currently 170 molecular methods for diagnostic purposes in the US. And of course, there are more, maybe over 1,000 in Europe that are CE marked. And we have to be careful because CE marking for this type of product, SARS-CoV, is not considered as a like a high stringent diagnostic test like HIV or hepatitis C or hepatitis B. So the manufacturer can do self-certification, but having said that, it has to meet the requirements of the European community. So it still has to be registered with the, they call it the competent authority, which is the equivalent of FDA in the US. And of course, in the Philippines, I have looked up their website, and they do recognize FDA authorization and CE marking or TGA from Australia or Japan, but they do have other additional requirements, like they would like to do validation using their own panels. So a manufacturer has to submit like 300 tests, for example. And so for this slide just gives a summary of what do you need to have to submit for authorization or approval. So you have to have your instructions for use, where you would describe your intended use, what is the limit of detection of your assay, analytical sensitivity to detect other strains of SARS-CoV-2. So as I have mentioned earlier, this virus tends to have or to show high frequency of recombination. So there might come a time, or it would come, a time would come where some of the current tests may not be able to detect the original strain of SARS-CoV-2. And you have to show analytical specificity by testing it against microorganisms which are well characterized. And also you have to test interfering substances. So for example, people who show COVID symptoms would take like Vicks Vaporub, right? Or other take lozenges or some other medication which would at least alleviate stuffy nose or all those symptoms. So you have to make sure that your kit uh, does not show cross reactivity or false positive results in the presence of those interfering substances. And most importantly, if you are able to do a clinical trial, which in the case of SARS-CoV-2 tests at the beginning of the epidemic, it was still an epidemic, there were no clinical samples. So as I have mentioned earlier, most regulatory bodies allowed blasting or comparing the sequences of your primers against a database of all possible microorganisms that could potentially interfere with your assay. And it's also important to show the stability of your kit at certain storage temperature or humidity. So you have to submit all this to be able to get authorization or approval. And again, this was waived for SARS-CoV, but most of the technologies, we already know the stability or the temperature requirements. So they took it as a guidance for manufacturers. And but having said that, they still would like you to put your kit on stability and come back to them and say that we have done stability study for our kit at different temperatures, and here are the results. So there is an oversight by regulators. I think the Philippine Department of Health also tried to reassess or evaluate the tests that are being used in the country. And so it's the same thing here and also in other countries. And there is a link in the FDA where they have made official the report on assessment of different tests. I know in the Philippines, they send out proficiency panels to certify labs and also to assess to perform quality assurance testing. Next slide, please. And so I have mentioned earlier that the culture collections played a very big role in the development of the tests. For example, there was no reference material except one in Italy at the beginning of the epidemic. So everybody used that, and then now they have more like ATCC or the BEI, DSM, and other culture collections are now advertising that they have reference materials for SARS-CoV and analytical specificity. So we had to order different kinds of microorganisms, viruses, bacteria, and one yeast, which is Candida, just to make sure that our primers do not cross-react with this. Although it's very obvious because they're totally unrelated, right? But regulators would always like to see proof. And then we did some clinical sample evaluation also, either using archived samples which were maintained by culture collections here in the US. So that's another function of culture collection, not just pure cultures, but hopefully there will be a repository for seroconversion panels or from different clinical specimens, which are of course well characterized. And then the next slide, I have mentioned that culture collections should be able to provide support to manufacturers as repository for strains that they use in production. It's always a good practice for manufacturers to deposit their strains not just in one collection, because if something happens to one culture collection, there should be a backup. Otherwise, that will be the end of their business if they lose their precious microorganisms. For example, reverse transcriptase is being produced by two viruses, and Taq polymerase by recombinant E. coli, for example. So there is a very big or important role of culture collections in safekeeping or trying to keep the cultures stable. I think I would like to end my talk here, and I would like to again thank the organizers, and please keep safe, and I'd be happy to answer your questions.
Thank you very much, Dr. Nadala, for that very interesting talk. Before we start with the question and answer, there's a request from the participants if it's possible to get a copy of your PowerPoint presentation. Is
Yeah,
it possible, ma'am?
I sent it to Carlo. Uh, I'd be happy to share it with everybody.
Okay, thank you very much. Okay. So the first question is coming from Ma'am Minette Iloreta. What is the mechanism of rapid antigen swab test that it will take minutes for its result? Is it reliable?
Uh, uh, as I said, for antigen swab testing is quite challenging because it would require tons of your of your virus, because most of the antigen rapid tests do not have the amplification of the target. So most of the antigen tests rely on uh, amplified detection, but not amplifying the target. So you should have a very, very good base technology to be able to detect the antigen from the swabs.
Okay, thank you very much, ma'am. There's another question. What is the advantage of using NASBA in comparison to RT-PCR? This is coming from Miss Rochelle Tuazon.
Yeah, both are good technologies, but as I said, if you're, for example, if you have very limited equipment or resources, with PCR you have to have like different temperatures, right? So in the beginning, we had to have three heating blocks, for example, for one high temperature, and then the middle, and then another heating block for another temperature. With isothermal, there's only one temperature. So you just need to amplify at a single temperature. So in terms of if you have manual um, equipment, not automated, then NASBA would be a simpler technology.
How about the cost, ma'am? How is NASBA compared to RT-PCR? Because it's normally one of the limitations. Like in the Philippines, a lot of people are not really into RT-PCR because it costs a little bit higher than the
Yeah, I
antibody
think
test,
that
for
the
instance.
limitation of the RT-PCR is you would have a separate set for extraction, right?
Mhm.
Then you have another set for amplification. Then you have another set for detection. So you need a big capital outlay. So you have very
Mhm.
complex instrumentation system, which would require preventive maintenance, and most importantly, it would require cold chain storage, right? So most of these enzymes require -80 or -20 degrees. NASBA or SAMBA, which is our test, is you. I was about to show you a short video of 2 minutes, but I could not share my slide. So NASBA is you put your sample and then you wait for 86 minutes, then you get your result. Nothing else that you need to do. So you just put your sample, put the 4 cartridges, and then push the button start, and then go do something else, and then come back after 86 minutes. If it's positive, it will already show a signal. But if it's negative, you have to go all the way to 101 minutes, which is about an hour and 40 minutes. And it's very ideal. Right now, it's being used in the UK in schools
Mhm.
because they don't have highly skilled technicians. So the nurse can do it herself, or the caregiver, or whoever is not really technically skilled, but it can be run by somebody without a high skills.
Okay, thank you very much, ma'am. The next question is asking for your expert opinion. Is the testing capacity of the Philippines enough for this pandemic?
To be
It's a
honest,
little political question. You may or you may not answer the question, but
no,
he's curious about
I
your
know.
own expert opinion.
I know. I have personally experienced, personally meaning my uncle who is in Surigao was tested positive by rapid test, right? Then he was told to be quarantined in like 3 hours away from where he lives. He lives in a very remote village. And then when he heard that he was going to be sent there, he was told that it was full. So it's overcrowded. So I had to talk to the mayor to explain to them that you're putting them more at risk. And so then he had to be swabbed. And then the results would take like a week or so. So I think it's not enough.
Okay.
Yeah. In
All
short,
right,
not enough.
that answers the question. Okay, there's another question. Are the mentioned techniques and or molecular diagnosis registered and approved by FDA? Do they need to undergo FDA's evaluation, registration, and approval before they can be used
In the Philippines.
in the Philippines? Yes, po.
Yeah, based on the website, they would they would take in all the certificate or the registration from other countries, but they still want to have 300 tests for evaluation in the country. So that would delay the implementation of other technologies in the Philippines.
Okay. There's another question. How do we do performance evaluation of the different COVID-19 detection kits? Example, clinical evaluations and determination of limit detection.
So, for the limit of detection, you should have a well quantified reference material. For example, a SARS-CoV RNA or an inactivated virus, which you can get from culture collections here and in Europe. And then what you do is you do different dilutions of the virus. You know what is the copy number per mil of the virus in your reference panel. So you run this panel in using the different tests, and then you would be able to see which one performs better or which one would have the best LOD or limit of detection. So you have to do a series of dilution. Then you find out the range, and then once you know that it's, for example, 1,000 copies per mil, you run that panel member 20 times, and then get to make sure that it shows 95% probability that it will always be positive at that limit.
Okay.
They can email me more if they want to have protocols or anything like that. I can help them with validations because that's what we have been doing for our assays.
Okay, so we'll be posting the email address of Dr. Nadala for those who would like to answer,
Mhm.
to make more questions. We're down to our last question. This is from Junel Julian. what is the best test for to use for detection of the SARS-CoV-2 in terms of reliability and cost reduction?
That's a very challenging question. I know I don't know how much it costs in the Philippines per test for molecular test. I heard sometimes it's
Around
like
5,000
6,000.
pesos, I think.
Huh?
5,000, around 4,000 to 5,000 pesos. I
Yeah.
think. I'm not also sure.
So I think with all the tests that are available in here, for example, like I would like to bring Samba to the Philippines, but I get discouraged because of all the additional hurdles that we have to go through, and then also maybe if you don't know anybody there, it takes longer to get the attention of somebody. So I would like to, for the Philippines, the ideal test would be something that you don't have to invest in a lot of equipment. You can have the point of care assay where you only put your sample, the cartridges, and then leave it there, and then wait for the test result in 1.5 to 2 hours. I cannot understand why it takes 2 weeks for the result to come back. And I know they have said that it has already shortened to maybe 3 days. I'm not sure,
Mhm.
but at least a week. But in the provinces, it's still very long.
Long.
Yeah.
Okay, thank you very much. We wish we had more time for more questions, but at this point, we would like to present our certificate of appreciation to Dr. Lourdes Nadala for gracing us this morning in our 20th annual symposium of the Philippine Network for Microbial Culture Collection. Again, thank you very much, Dr. Nadala, for your time.
Thank you, and mabuhay ang Philippine Network of Microbial Culture Collections.
Okay,
Hope to
thank you.
see you in 10 years.
Yes po. You will be our speaker in our 30th year celebration.
Thank you,
Thank you very
Joel.
much, Dr. Nadala. Okay, so we are up to our last speaker for today's webinar, for this webinar series. Our last speaker, but certainly not the least, is Dr. Christian Mejia Pastor. Okay? Dr. Pastor finished his Bachelor of Science in Chemistry for Teachers at the Philippine Normal University. He then took his MS-PhD program in Biochemistry at the University of the Philippines, Manila, under the PCHRD DOST Scholarship. His current, his current post includes being a principal investigator of SHED-DARE2 program, a non-invasive determination of saliva-derived proteins as biomarkers of stress manifested in electrocephalographic imaging. He is also the director of capacity building program in science and mathematics education for DOST SEI graduate scholars and a Math and Science Cluster Coordinator of the College of Graduate Studies and Teacher Education Research of the Philippine Normal University. From May 2011 until present, he is teaching full-time as a faculty of chemistry at the Philippine Normal University. Among his work experiences, it spans from the academe, either as guest lecturer on foundations and applications of biotechnology and molecular pharmacology, or mostly as a trainer or facilitator in K-12 national training of grade 9 teachers for science in the National Capital Region. He is also very active in research, and his research field of interest mostly is on multi-drug resistant Mycobacterium tuberculosis isolates. Okay? Dr. Pastor has presented a lot of his papers in several workshops and seminars and conferences, not only local but also abroad. Dr. Pastor is also an active member of several scientific organizations such as the Philippine Society for Biochemistry, and Molecular Biology, the Philippine Society for Chemistry Teachers, Biology Teachers Association of the Philippines, and of course, the Philippine Network for Microbial Culture Collection. Ladies and gentlemen, let us all welcome Dr. Christian M. Pastor, who will be talking about COVID-19 testing, rapid testing of COVID-19. Ladies and gentlemen, Dr. Pastor.
Okay, so good morning, everyone. Thank you, Dr. Cornista, for the introduction, and I also would like to thank the organizers of PNMCC for inviting me to share some of the concepts about antibody and antigen tests for COVID-19. Okay, so I would just like to ask first if you can see my slides. Can you see them now? Yes. Okay, so I'm going to talk about the science behind rapid tests for COVID-19. So I would also like to thank Dr. Nadala for giving the or providing the necessary all the necessary information. That's a good backgrounder for my topic, simply because most of what Dr. Nadala said or mentioned are actually important in understanding the different rapid tests for COVID-19. So without much further ado, I would like to provide you the outline of discussion or the things that I would like to share with you this morning. The first is, of course, just to have a certain background about what's happening right now in terms of the COVID-19 pandemic situation in our country. Next is proteomic targets for SARS-CoV-2 detection, because most of the time when we are talking about antibody and antigen tests or tests or diagnostic assays, they are relying on proteomic targets or protein targets. The next is the basis or the basics of antibody, antigen, and their interaction. So that will be quite short because most of us, if not all, are familiar with this concept. And then we're going to look at some of the antibody and antigen tests for COVID-19 and what are those kits that are being used in the Philippines, how do they work, and let's look for future directions and challenges regarding this kind of test. Okay, so let me just give you a certain background about history of pandemics. Dr. Nadala has already mentioned that there has been so many pandemics. Years ago, the Asian people suffered it, okay, and there are so many pandemics that the human population has already met. And we are hoping, but we cannot be so sure that after this SARS-CoV-2, there will be no more. Okay, so some of the most remarkable pandemics, you see there Black Death or the bubonic plague. You also see there the other great plagues, and what triggered the production of most of the antibodies and vaccination movements is the smallpox. Smallpox, which killed about 56 million people. And after that, we have the cholera outbreak and yellow fever, and then we have HIV, and then by this time we have COVID-19. So in terms of the death toll, of course, among the viruses and all of these plagues, Black Death has killed the most. Okay, but as we can see, the in terms of the number of deaths, of course, HIV would also rank on a higher part or level. And then we have here COVID-19. As of now, on the next slides, I'm going to present to you the statistics according to the World Health Organization. So let us look at closely on the different properties of this virus. SARS-CoV-2 is different from COVID-19. So we are familiar with this, but we have to reiterate that the SARS-CoV-2 is the virus and COVID-19 is the disease. Both of them were announced as official during February 11, 2020 by the WHO. And as of now, almost everybody is familiar with this thing. Okay, and in terms of the series of events, as early as December 31, 2019, they have this news and SARS-CoV-2, but they are not aware of what it is. They are just observing viral pneumonia in people in Wuhan, and then after some time, we know the story. It arrived in the Philippines like by month of February, wherein we have the first confirmed case of a Filipino citizen who was infected by SARS-CoV-2. And February 11, WHO announced that the disease caused by this coronavirus should be named COVID-19. So along these events, there are so many diagnostic kits that were developed, and they were still being improved as of the moment. Okay, so one of them is RT-PCR, and after some time, they also developed the antibody and antigen-based test for COVID-19. Okay, so in terms of the number of cases, so there are already 34 million confirmed cases worldwide as of yesterday, and there has been 1 million, about 1 million, more than a million deaths due to COVID-19. And in the Philippines, we had 314,000 confirmed cases and 5,562 deaths. So all of these statistics are actually increasing after some time, and we hope that things would get better. Um, the diagnostic kits will be made, the different treatments could be made, but I'm sure that it will take some time. We have to wait for a certain period of time for us to do them or to prepare them. So in terms of taxonomy of coronaviruses, the COVID-19, the SARS-CoV-2 that causes COVID-19 is actually a beta coronavirus. So it is together with MERS-CoV, SARS-CoV, the first thing that we encountered, a year or 2 years back. And these are actually infecting or they are capable of infecting the lower respiratory tract as compared with other types of coronaviruses. This is what I got from a journal, that they are proposing that after species, there should be an individuum level, so that the classification or the levels of classification for that virus will be more clear. And let us now look at the genomic structure, because whenever genes are expressed, proteins are produced. And when these proteins are produced, they could be good targets for antibody and antigen tests. So of primary importance are four main genes: the gene that codes for the spike protein, the gene that codes for the envelope membrane, and the nucleocapsid of the virus, and other miscellaneous proteins, which all are also important for the virus to replicate and exit the cell, is given in the table. So despite the fact that they are not that much of a target for antibody testing or antigen testing, they are also important because they could be a potential target for drugs. Okay, for designing of drugs for COVID-19. Okay, so let us now look at these four basic proteins that I mentioned. So one thing is that when you get a cross section, this is just, of course, an art, a sketch or an illustration of how the virus would look like. And one of the most important part of the virus is said to be the spike glycoprotein. The main role of the spike glycoprotein, as you can see in your screen, all of those characteristics are given, but the main mechanism or the main importance is that it is the one that interacts with the ACE2 receptors that are found in human cells. And the spike protein is actually composed of, it is a heterotrimer. It means that for it to function, there should be a group of 3 of these proteins. And aside from that, each segment or each monomer can be cleaved into 2. So it has an S1 and an S2 fragment. So those 2 fragments are playing different roles. The S1 fragment is the one that interacts with the receptor, and the other one contains the fusion peptides or fusion portion of the spike glycoprotein, wherein once that the S1 is cleaved, the S2 portion of this spike protein will initiate events, mechanisms to for the virus to invade or enter the cell. So these are some of the depictions uh, of the protein, the spike protein. The spike protein is said to be trimeric, as you can see there, and the white ones are corresponds to the receptor binding domain, or the RBD. The RBD is the target of most antigen tests and antibody tests. And when you look at some other depictions of the protein, so as you can see on the lower left, it the journals would somehow tell us that the among the three, there is only one that should be on an up conformation. And once that it is on an up conformation, the other two would be on the down conformation. So yung dalawa naka-fold, yung isa nakaangat, at yung nakaangat na yon na part ng RBD or ng receptor binding protein is the one that interacts with the ACE2 receptor. Okay, so in this slide, sorry for quite small figure, but it is an alignment of the different spike proteins of the different coronaviruses. How is it important, or why is it important in development of tests? The specificity of the test relies on the ability of the antibody to search for its target. And when you look at before we can design a certain specific test for SARS-CoV-2, we have to ensure that the target, the epitopes or the amino acids that serves as the target of our antibodies is unique for this SARS-CoV-2. So if the sequence of amino acid can also be found in other coronaviruses, hindi magiging specific yung test. And more so, if those stretches of amino acids that you choose is found in other organisms, cross-reactivities will also occur. So there will be false positive results. Okay, so that somehow is the way that we will present the next slides. So when we look at the other proteins, the membrane protein is one of the highly conserved protein in the SARS-CoV-2 virus structure. So most structurally ordered and maintains the shape of the viral envelope. And it is also important for assembly of the protein or of the virion once that it goes out of the cell. And if you're going to look at the alignment of the proteins that correspond to the membrane protein of SARS-CoV-2 as compared with other coronaviruses, we can say that it is quite conserved, and then that's why that somehow explains why there are few to none na antibody and antigen tests that will attempt to detect it, because hindi siya masyadong specific for SARS-CoV-2. When you look at other proteins, for example, other segment of that protein or other part of the protein, they are almost the same with other viruses. In terms of similarity, the first SARS-CoV virus that our population encountered, and the SARS-CoV-2 that we have right now is 79% similar. But it is not very similar when you compare it with the similarity between SARS-CoV-2 and those coronaviruses that are found to infect bats. But the coronavirus that infects bats and as compared with the SARS-CoV-2, they are 98% similar in terms of their genetic makeup. So let us proceed to the next. So aside from that, we have the envelope protein, and it resembles a pentameric assembly. Okay, and this envelope protein is very important because most of the internal functions of the virus is mediated by this protein, as well as the other accessory proteins that I mentioned a while ago. And this envelope protein is also highly conserved. As you can see, there are few amino acids that are quite unique for this SARS-CoV-2 when you compare the sequences of its proteins, the envelope proteins, with other coronaviruses. So I would like to reiterate that when we are looking for targets, we are looking for stretches of amino acids that are unique for this virus, so that we can somehow differentiate it or discriminate it from others. So that's it. And lastly, we have the nucleocapsid protein. So it binds to the RNA genome, and it facilitates the encapsulation. It has also the ability to self-associate. And actually, the nucleocapsid protein is a target also for antigen and antibody testing. Okay. So in one of the journals that I found here, it might be very busy, but it simply tells us that whenever the virus enters the cell, it somehow of course, we are expecting that it will replicate. But after replication, they are saying, or the journal is suggesting that there will be what you call pyroptosis. Pyroptosis is a highly inflammatory process which somehow leads to the cell death. Uh, it is because of the response of the cell to the microbe, or just like in this case the SARS-CoV-2 virus. And after some time, what happens is that inflammation due to the secretion of pro-inflammatory molecules will occur. And somehow after some time, of course, the individuals will be classified into two. They could be healthy and their immune system can cope, and then that's why they could produce antibodies, and they might be a good source of convalescent plasma after. And some subset or a subset of the population might not make it because they have dysfunctional immune system. They might be immunocompromised or whatsoever, that somehow hinders them to create these antibodies that are very specific to the proteins of the SARS-CoV-2 virus. So for us to understand the timeline, because this is very crucial, and then others will be discussed faster. When we look at the incubation period and the infectious period of the SARS-CoV virus, we can somehow simplify it this way. So here is the timeline of a 30-day or an average of a month duration. And once a certain person is infected by the SARS-CoV virus, the incubation period, according to journals, is 2 to 4 days. But 95% of people infected will show symptoms on the fifth day. But for us to simplify this figure, let us say that after 14 days, dun pa lang nag-emerge yung symptoms. When a certain person got sick after 14 days, a mild and severe duration of COVID-19 might be observed. It depends on of course, on the person, kung yung immune system niya could take it or not. But we have to be very cautious, because they also said, or they also mentioned that 2 days prior to the first day wherein the symptoms were observed, they are highly infectious. They are highly infectious. Now, let us put this diagram in a perspective of how the different tests will give you a positive or a negative result. Uh, when we look at the timeline and extend it to like 2 months, okay, let us extend it to 2 months. The first thing that you're going to observe is the rise of the viral load, and that's why when there is a rise in the viral load, you can get them or you can isolate them from the respiratory tract. Therefore, antigen detection through kits can be done. Okay, they can be done, and they will give you somehow a positive result. And they tend to approximate the RT-PCR results. And but of course, in terms of sensitivity and specificity, we will go for RT-PCR because that is the gold standard. Now, the second line here that appeared in our graph is the nasopharyngeal swab RT-PCR, and therefore, as you can see in the figure, there is a time wherein the RT-PCR would favorably give you a good result. But as a person recovers, of course, what happens is that the viral load declines and the RT-PCR might be negative. Now, the strength of the antibody and antigen test might be more of the latter part of the story. So you have here the IgM that can be detected in the blood. On the early phase, some of the journals would say within the first week it is being developed, on the middle of the first week, on the third day, more or less. It depends on the journal that you're reading. It depends on the data that they got. And after some time, you have the IgG. Of course, the data here it might not be exact because it is just to illustrate the relationships between these variables. And when we look at the antibody detection, IgM and IgG will somehow give you a certain leverage in terms of when you are talking about this part of detection. Okay, so as you can see, IgM, the level of IgM drops after some time. IgG, if we are looking at immunologic memory, then IgG might do that, but there is a limited study which tells us how long will those immunoglobulins or Igs will stay in the blood. So this is what I got in another journal, and somehow it gives us the idea and it extends the timeline for a year. Okay, so let us look now at the antigen antibody chemistry. An antigen is anything that binds to your, or that is being recognized by your antibodies. And the part or the part of the antigen that binds directly with your antibody is called the epitope. The epitope consists of amino acids that you chose, for example, in eliciting that immune response and creating that antibody. So that's it. And of course, in three-dimensional structure, we can see it as this. And there's another way of looking at it, in which, as you can see here, the orange ribbon structure is the SARS-CoV-2 spike receptor binding domain. So that is in most of the tests that I scanned, that is what they are targeting for detection. And I would like to reiterate that the part of the SARS-CoV-2 spike receptor binding domain that interacts with your antibodies is called the epitope. In the laboratory, you can use bioinformatics softwares to choose that epitope and test your antibodies if they could detect the presence of SARS-CoV-2 virus or proteins in your sample. So let us look at some other concerns. Last of the concern here is cross-reactivity. So let us take note that there are times that even if we design antibodies and we choose antibodies that are unique in terms of their amino acid sequence, the folding of those amino acids may also resemble other folding patterns of other epitopes of different sequence. So kaya ito nakalagay dyan, an antibody may react with two different epitopes, and somehow we would like to minimize this in terms of when we are looking at diagnostics. Okay, so this is, let us now look at the different kinds of tests. Meron tayong tinatawag na point of care at saka yung mga ELISA-based na mga tests. So this video will somehow summarize how it works. Okay. So when we look at these are they resemble or they are like pregnancy tests, right? Test. Okay, so they work using the antibodies that are specific to the COVID, the antibodies that recognize the antigens of your COVID-19, the IgG and the IgM that is being produced or that are being obtained from the blood of the blood sample. And then we have here the human anti-human antibodies, which serve as the positive control. So when we are interpreting this kind of tests, ang mahalaga dito is that laging meron nagwo-work yung positive control. So whenever the positive control is not working, so somehow it invalidates the result of the test. Okay, so as we can see in the graph a while ago, it is also there is a possibility that you can detect M only, G and M, but seldom G without M, or otherwise G only. It depends on the phase of the of the infection or of the disease, or the state of the patient. Okay, so there is also what we call ELISA. So most of us are familiar with this, and we know that it uses enzymes, and then there's a substrate that produces the substrate that you give to the enzyme is being processed to produce a colored product. But the amount of enzyme that are being that binds to to the wells depends on the amount of antigens that are present in that well. And the amount of antigens depend on the amount of viral load that is present in the sample. Two major types of ELISA that is being used are direct and sandwich. Okay, so let us now remember some things. In SARS-CoV-2 infections, IgM and IgG antibodies can arise nearly simultaneously in serum. And but there should be more studies on how long IgM and IgG antibodies will remain detectable, and how long they could protect you from SARS-CoV-2 or COVID-19. It is also important to note that some persons do not develop detectable IgG or IgM antibodies. So there are some studies that mention this. So it should not be surprising that that we cannot find or get some results out of these tests. Kaya nga yung sinasabi ni Dr. Nadal kanina na parang in terms of sensitivity, mas maganda pa rin na gumamit tayo ng RT-PCR as compared with these kinds of tests. Okay? So let us now look at the rapid test and let us compare the two. A viral test or an antigen test used to check the samples from the respiratory system. It could be a swab, nasal or oral swab, and it is used to diagnose acute infection of both symptomatic and asymptomatic individuals. In the FDA website, they have protocols and guidelines for this. But when you are talking about antibody test, it is a serologic assay. So you would like to survey or you would like to conduct an epidemiologic study to understand the transmission dynamics of the virus. And it may support clinical assessment of persons who present late in their illnesses, in conjunction with other viral detection tests. Now, let us look at the status. The FDA has granted emergency use of the authorization of antigen tests, but FDA has not authorized using antibody test to diagnose SARS-CoV-2. Hindi maganda siya for diagnosis kasi late lumalabas yung antibodies. The CDC does not currently recommend using antibody testing as the sole basis for diagnosis. And our government is also giving some guidelines that runs parallel with that or consistent with that. So let us look at the antigen tests, advantages and disadvantages, and we will just move across this quick. So the major antigenic targets will be SARS-CoV-2, not the live virus, because of course coming from the sample, they have to process it first. Rapid turnaround time, relatively inexpensive. They can be used at the point of care. They perform best when the person is tested in the early stages, but not on the late stages. may be informative in diagnostic testing situations in which the person has a known exposure of a confirmed case of COVID-19 and can be used for screening testing in high-risk congregate settings. For example, people who are living on a certain area na crowded sila doon, okay, like dormitories, ganyan. So disadvantages, it is generally less sensitive than viral tests. It is really less sensitive as compared to RT-PCR. And there is a limited data on the use of the rapid antigen test as a screening test for asymptomatic patients or people. Okay? And it may be necessary to confirm a rapid antigen test result with a nucleic acid test. Of course, that would be our RT-PCR. And then antigen levels in specimens collected beyond 5 to 7 days on the onset symptoms may drop below the limit of detection. So therefore, there might be some problems in, you have to get fresh samples. The proteins can be denatured after some time. Nasisira din yan after some time because there are some enzymes that could cleave them and that will affect your results. So this is somehow the a summary of the differences between RT-PCR and antigen tests. Now, let us remember, these are things to remember. In most cases, negative antigen diagnostic test results are considered presumptive, and they won't tell you that you certainly have or don't have COVID-19. When used for screening tests in congregate settings, presumptive lang yan. Kailangan meron tayong verification with RT-PCR. Okay, that's it. Now, let us look at the other test, which is the antibody test. What are the advantages? Of course, when we say that antibody test cannot be used for diagnostics, so para saan siya? Okay, so it also has different another set of contribution, okay, or importance. In terms of the basic considerations, let us remember that IgM is most useful for determining recent infection because it rises in our graph a while ago earlier as compared with IgG, but IgG will just give you an idea of a long-term immunologic response that you have been infected before. That is what IgG will tell you. And IgA found in saliva, not necessarily in blood. So rapid turnaround time, about 30 minutes. It helps you determine whether the individual being tested was previously infected. And it could detect resolving or past SARS-CoV-2 virus because it somehow, those antibodies somehow appears after or in the latter part of the infection phase. And it can aid in determining who may be eligible for donating convalescent plasma so that others can be can be cured as of now kasi wala pa tayo talagang drug or vaccine against it. And one of the advantage of antibody test is that it provides demographic and geographic patterns of who among the population have developed the immune response against this virus. So some of them, hindi nila alam na sila ay infected, but they didn't show the manifestation of infected. So through this test, they will know. The disadvantage is that the first one, currently there's no identified advantage for this. And that's why it is not recommended for us to use this for diagnostic purposes. Serology tests can yield negative result even in infected patients. It depends on the time element, of course, and it might be false positive. So if there is a novel strain or maybe a variant that somehow the mutation strikes that area which is the target of the antibody test, then your result will somehow become false positive. And additional data are needed before modifying the health recommendations based on serologic test results. Both WHO, CDC, FDA, and even in the DOH is saying the same thing. So let us look at some of the, in terms of the key points, positive results from appropriately validated serology tests that are very specific for SARS-CoV-2 can indicate whether a patient has a recent or has had recent or prior COVID-19 infection. But you do not use that for diagnostics. Okay? So what are the future directions and challenges? Generation of neutralizing antibodies. I have read so many researches that are attempting to develop neutralizing antibodies. These are antibodies that can somehow block the action of the virus so that it cannot infect the cell anymore, or it cannot go out of the cell anymore. So it somehow impedes the viral entry or exit so that other cells won't be damaged further. And there are so many mutation studies. I'm sure that some of us are familiar with the GISA-8 database, and they are looking for mutations that might somehow affect the diagnostics, the antigen and antibody tests after some time. So in the Philippines, there are about 260. This is as of September 29, 30. FDA has released a total of 260 COVID-19 test kits. 89 of them are PCR-based, 93 of them are antibody tests, and 58 of them are immunoassays and other categories. So you can access this website for you to get the full list of those test kits, the names of the test kits. But I would also encourage you to update. You just have to visit that website from time to time because they are revising the list. And there are also some licensed testing centers. So not all testing centers could be licensed. They have their guidelines for this anyway. So I would like to say that I found a document dated March 31, 2020, wherein there is a circular that gives guidance on the use of COVID-19 rapid antibody-based test kits. And they said that I would like to highlight number 2, all rapid antibody tests cannot be used as a standalone test to definitively diagnose COVID-19. RT-PCR kits remain to be the gold standard. And they just gave some other guidelines, additional guidelines in terms of whether IgM is positive, IgG is positive, what you should do. Okay? So this document is also accessible from the FDA website. And I would like to also highlight number 3, the rapid antibody tests, most of which are IgM and IgG can be used as an adjunct tool. And then for number 4, letter A, the rapid antibody test kits cannot be used for mass testing or self-testing, over-the-counter, or self-testing. So please, we have to be aware about these guidelines and rules so that we may not violate them. No? Kasi yung iba, they are really interested whether they are infected with COVID or not, despite the fact that they don't display some symptoms. Okay, so lastly, I would like to everyone to watch this video. This is coming from the genomic epidemiology of SARS-CoV-2, from the GISA-8 database. And somehow it gives us the idea of how the virus mutated or the different variants of the virus emerge as it travels from one place to another or one continent to another. Sorry. So hindi yata nag-work yung hindi yata nag-work yung video. So ayan. So please watch this one first. So as you can see, the graph that they had started from Wuhan, China, and then the transmission of the virus has been tracked because of the genetic sequences of the virus. Some of you might be interested in asking, we are talking about proteins here, so why are you showing genomic sequences? It is because when the genomic sequence of a certain organism, or in this case, hindi naman regarded as organism, a virus changes, it also changes the profile of the proteins or the proteome of that virus. And whenever those protein targets are changed, then our diagnostic kits might have a problem. And it will also affect RT-PCR primers because most of the primers, not most but all primers, are based on the sequences of these viruses that are available in the databases. So you might be familiar with some news two months, 3 months back that there is an emerging, uh, they said strain, but it is a variant of the coronavirus that is more deadly because it has a mutation in the spike protein. As early as March, that variant of the SARS-CoV-2 has already appeared in the GSAID database. But more studies must be done for us to verify whether that mutation somehow confers an additional advantage to the virus or in terms of its infectivity. Okay? So, fast track natin yung video. Sorry. So, when you look at the outcome of the video, it looks like this. And each dot there corresponds to a country, a sample from a country. Okay? So, you can visit the GSAID database for that purpose. Okay, so that's all for rapid test for COVID-19. Thank you everyone for listening.
Thank you very much, Dr. Pastor, for that very comprehensive presentation. Again, a lot of our participants are amazed with your presentation and they are asking if it's okay to get a copy of your PowerPoint.
Opo.
It's
I
great.
could share the PowerPoint because almost all of the contents were obtained from journals and some agencies like WHO, CDC, DOH. So these are public documents.
Okay, thank you very much. Okay, so there's one question from Dr. Marilyn Bololong, resident microbiologist of UP Manila, your alma mater. Okay, the question is, which is more protective or may be responsible for the pathogen clearance in COVID-19 cases? Is it IgG or IgA being a mucosal pathogen?
Okay. In terms of mucosal pathogen kasi ang protection ng IgA. So there are that it might be a potential it could potentially protect an organism in terms of, for example, a human for against SARS-CoV-2. But in terms of ano kasi ma'am, pagdating sa memory, mas well established yung IgG. So and when the virus comes into the body and it somehow bypasses the mucosal barriers. That is what we observe. The IgG would have a greater role in terms of dealing with that virus. So IgA might be more of before it enters. It's good that we have IgA there in the mucosal as a part of the mucosal barrier. But of course, the virus will always find a way to invade cells inside. So the IgG might be better. That's it. Pero mas maganda sana kung silang dalawa ay present to protect us against that virus.
Okay, thank you very much. There's another follow-up question. What is the probability of cross-reactivity when lateral flow-based assays will be used for SARS-CoV-2?
Okay. Also kasi naglabas ang DOH, FDA ng di ba ng list, and I tried to look at their product inserts. And this is one caution for this might be a caution for everybody, Now when you are looking at when you subject yourself to a test, tignan niyo muna mabuti yung product insert or specification ng testing kit na ginagamit sa inyo. Yun nga yung sa pagdating sa cross reactivity, we are saying that the antibodies, especially if they are monoclonal, specific naman yan, specific yan para doon sa virus na tina-target niya, like this in this case SARS. We have to look at sensitivity, specificity issues. And somehow yung meron din tayong tinatawag na positive predictive values, negative predictive values. So in our biostatistics we encounter those terms. And so we have to consider those things. And aside from that, I've also found that there are some antibody tests that in their product insert, that the antibodies that we have here cannot discriminate between SARS-CoV-1 and SARS-CoV-2. So nakalagay yun sa product insert, pero syempre pag binenta yan sa di ba? Pag ginamit yan in the healthcare system, pag binenta yan sa market, hindi naman tinitignan ng person yan kasi ang tinitignan niya is a band, positive or negative. So I would like to advise everybody to be cautious when we subject ourselves to these kinds of tests. Yun po. So I hope I answered the question. So kung
Okay.
monoclonal, ano naman, okay naman yung specificity, yung sensitivity and other parameters, then we could somehow trust that assay, that it will tell you that kung positive ka talaga. Kung wala kang wala kang COVID-19, negative ka. Kaya lang tignan natin yung mga product inserts kasi they will give you a better detail. And I'm quite surprised that not all product inserts of all of those diagnostic kits are giving us the complete set of information that we would like to know. So makikita nyo yan kapag tinabulate ko yung lahat ng information. So some of them might be very complete, some others might be for proprietary reasons they want to disclose. Ayun po.
Okay, so due to time constraints, we will have our last question. I think it was already answered previously, but nevertheless, I'll gonna state it anyway. Do serological tests for SARS-CoV-2 cross-react with other coronaviruses? How can we rule out cross-reactivity between other deadly coronaviruses such as SARS-CoV-1 or the Middle East Respiratory Syndrome coronavirus?
Okay, so it's quite a tricky concern kasi pag tinignan natin at the very least we are looking at protein sequences. When we access the laboratories, in the laboratories we have your softwares. Anyway, kahit naman hindi sa lab, pwedeng i-access yan. For example, the GSAID database, all of those protein sequences are actually available. When you compare all of those sequences, makikita natin na itong mga part na to unique, itong mga part na to hindi. Siyempre, pag hindi sila unique, hindi sila unique sa SARS-CoV-2 virus, you cannot use them for di ba, parang discriminating between which is SARS-CoV-2 and which is not SARS-CoV-2. I also mentioned a while ago that, but apart from that, we really have to test it. We do not have to trust the sequences only na parang, ito unique sa SARS-CoV-2. Magbibigay ito ng specific na result. Hindi. Kasi there are times na kahit iba yung amino acid sequence, when they fold on a three-dimensional structure, they also have almost the same folding. So that's another danger. And then that's why the laboratory tests that are very important in terms of testing specificity of certain tests. I found some product inserts that they enumerated almost all organisms that they tested, whether the kit that they are introducing to the public will somehow give some cross reactions with the proteins that are found in other organisms and in other coronaviruses. So pwede po yun. Tignan nyo lang po yung product insert. Masaya ding mag-explore actually na pag tinignan mo yung mga product insert nila, makikita mo some of them might cross-react with influenza viruses. So somehow, that gives you the idea, yun yung parang disclaimer ng product na, oops, hindi ibig sabihin sinabi ko na positive yan, meron pa ring ibang dahilan. Actually in the FDA guidelines, meron nga sila pang parang nabasa ko rin na just in case that you really cannot rule out what's happening in the test, you have to refer the patient now to an infectious disease specialist. Maybe because the test might give you weird results because the pathogen that's infecting the patient is not necessarily SARS-CoV-2. Kasi marami ring general na symptoms and manifestations of SARS-CoV-2 na hindi na nakikita rin natin sa ibang diseases. Nakakatakot lang ngayon kasi pag umubo ka, feeling lang lahat SARS-CoV-2 na agad yun. So, mali mo iba naman. So that's it. But the manifestation is almost the same or similar. Ayun po.
Okay, thank you very much. We wish we had more time, but unfortunately, we are limited by the time. At this point, I would like to present the Certificate of Appreciation from PNMCC to Dr. Pastor for gracing us in our first day of a series of webinars of the 20th PNMCC Annual Symposium. Thank you very much, Dr. Pastor. At this point, may I then give the mic back to our host, Sir Dino, for the continuation of the closing ceremonies of today's webinar. Sir Dino?
Thank you very much, Professor Cornista, and to our three esteemed speakers. At this point, we will be watching a product presentation from one of our partners, Yana Commodities. So please watch this. All right. So the Philippine Network of Microbial Culture Collections would like to present this certificate of appreciation to Yana Commodities for partnering with the organization to make this virtual seminar successful. Thank you very much, Yana Commodities. At this juncture, ladies and gentlemen, we'll be calling Dr. Anacleto Argayosa, the Department Head of Biology of Trinity University of Asia, to give us the seminar synthesis.
All right, so I'm sure you're ready for lunch, but I'm just going to keep this short. But first of all, I would like to thank our excellent speakers for really sharing their expertise on the topic. And let me start with our keynote speaker, Dr. Ida Dalmasio, for giving us the relevance of this culture collection, microbial culture collection, and its use. No, so indeed, culture collections are a good source of microorganisms as reference strains and a source of possible biotechnological applications, including the development of vaccines, no, and sources of these antigens. So it also, Dr. Dalmasio also included that this actually contributes to the millennial development goals in the world. And these culture collections are a good source of bacteria that can be used even not for vaccine itself, but for probiotics as our ability to combat infection via innate immune responses. No, so these diverse groups of organisms can be used indeed as a good bacteria to at least balance these onslaught of bad bacteria as well as a viral infection. So these probiotics might indeed be a good source of help and mitigate disease due to COVID-19. So she also mentioned that these microorganisms can be used as biosensors, no, with their enzymes and natural products as well. So thank you so much, Dr. Dalmasio, for your excellent presentation on the relevance of culture collections to our country and to the world for these applications as well. For our next talk, given by no other than Dr. Lourdes M. Nadala, so she talked about these SARS-CoV-2 testing platforms, no, for COVID-19 diagnosis. There is like a race, indeed, as I appreciated. There's been a race for vaccine development, and there's also been a race for diagnostic tests. So she reported that at least in the U.S., there are 170 molecular methods for diagnosis, and the gold standard still is the RT-PCR. Yet she mentioned that there are also issues with some of the tests that came out because of the standard to approve this for use. So there's a need for analytical sensitivity, for specificity, and for stability. So she also mentioned new techniques or new tests, such as these nucleic acid based isothermal systems, no? So which is seemingly less expensive compared to the gold standard RT-PCR, as well as the new one, the CRISPR technology, so which also can use the technology based on the isothermal amplification system, including the lamp. Okay, so these tests are being used, and some of them have been used in the world, no, extensively in Europe, in America, and in the Philippines. So she just also mentioned there's a need for these tests to be sensitive because some of these tests present low sensitivity, about 30+%, and this is not good. If you're screening the population of COVID-19 infection, so they might be tested negative, but they'll just be walking around with the viruses and infect the population. So that's about, I think I could remember from the talk of Dr. Nadala. And she excellently, of course, gave the mechanism of these tests from the RT-PCR to CRISPR. So thank you so much, Dr. Nadala. Well, the last one is an elegant lecture and presentation on the science behind this tests, no, rapid tests. So this was given by no other than Dr. Chris John Pastor, and I'd like to just show you that there are indeed many proteins in coronaviruses. Although the corona itself, the protein that's in the spike, seems to elicit this first antibody response, right, and is detected as the first epitope. There are other candidate. He mentioned that there are indeed other candidate proteins that can be used for more specific tests because the spike protein on the coronavirus, SARS-CoV-2, shares similarity in the sequence as well as in the folding with other coronaviruses, including the common colds, right? So it's a good insight that we could explore, or studies could be done to explore these other possible antigens or viral proteins in the coronavirus as a good and more sensitive test for these rapid antigen test for these COVID-19. So that's one, and the other one is I believe he also explicitly compared the applications of these antigen tests, which is rapid, as well as the antibody tests. No, so they have various applications, and also mentioned that. Including Dr. Nadala mentioned that this was approved for emergency purposes as an adjunct, okay, to the RT-PCR test, because this is rapid, so you can take this test as an indication of an active infection or recent infection, especially if the tests that have been developed is really sensitive to this antigen and specific. So it has that application. It was approved for these emergency applications for diagnosis, but with the RT-PCR as the gold standard. Now, the antibody test is also available, just like a dipstick, and it does shows a good application for screening those patients that have recovered from COVID-19 as a source of convalescent plasma and other um, applications on serologic and epidemiologic studies. So kudos and congratulations to our speakers. I will not keep you for long, and I'm sure you got a lot from these talks, and looking forward to the next webinar series of the 20th PNMCC Annual Convention and Symposium. Thank you very much.
Thank you very much, Sir Lito. And we will now be calling Professor Carlo Chris Apurillo, the PNMCC Vice President and Faculty of the Philippine Science High School at Eastern Visayas Campus, to give us the closing remarks.
Thank you very much, Sir Dino. Good morning to all of you. Today we mark the opening of the PNMCC's 20th Annual Symposium. The PNMCC was established in 1996, and its first convention was held in the year 2001. Despite the pandemic, we are able to successfully mark this important milestone. We are truly honored that Dr. Dalmasio and Dr. Nadala, two of the founding members of PNMCC, were able to join us today. We would like also to thank Dr. Pastor for sharing his expertise in today's webinar. 2020 is also the 24th year of PNMCC from the time that it was established. So we are gearing up towards our 25th year in 2021. We hope that next year we'll be able to meet each other face to face. Indeed, this is just the opening of our 4-week webinar series. Since we are marking our 20th annual symposium, the board of directors decided to do a month-long symposium. We still have 3 more Saturdays where our esteemed resource persons shall share their expertise in different topics along the theme 3 C's, COVID-19 and culture collections. At this point, I'd like to thank all of you for joining us today here in Zoom, in Facebook, and in YouTube Live. We hope that you will again join us in the succeeding weeks. For those who have not yet applied for membership, we have already opened new ways to pay for the membership fees. We are looking forward towards having you as new members or as regular members or even as lifetime members in this organization. As the Network for Microbial Culture Collections in the Philippines, it is our mission to assist our members in establishing your own culture collections. I would like to thank our partners at Trinity University of Asia College of Art, Science, and Education, headed by the Dean Dr. Howell Ho, who are tirelessly working with us to bring you this webinar. We would like also to thank our sponsors, Yana Commodities, who was featured today, ESCO, Diomed, and Lifeline for their support. And of course, the PNMCC Board of Directors headed by Ma'am Dayan for working hard despite the distance in order to hold this annual symposium during this pandemic. With that, again, I say thank you, but not yet goodbye. We hope to see you again next week for another round of interesting discussions. Please invite your colleagues and friends as we continue with our webinar series for the 20th PNMCC Annual Symposium. Maraming salamat po.
Thank you very much, Sir Carlo. And that concludes our program for today. Join us next week as we present the second installment of this four-part webinar brought to you by the Philippine Network of Microbial Culture Collections in partnership with Trinity University of Asia College of Art Sciences and Education and the Biology Department. Links to the webinar evaluation will be posted in the chat box and the comment section of Zoom, Facebook, and YouTube, and will also be sent to your email. This has been your host, Assistant Professor Dino Cantal of Trinity University of Asia, saying thank you, God bless, and stay safe and healthy. See you next Saturday.
Salamat po.